recording. How do I do this? There we go. Okay. So let's see here. Here's my list of. All right. First of all, let's start out. Well, while we're waiting on people to hop on before I get into genetic stuff, do you guys have university level questions, semester level questions?
Someone had posted earlier a notification they got from one of their professors saying that, like, you should hear something today about FMU and stating their pass-fail.
That professor should be roundly scolded.
Figure out who. I'll tell you who it was.
No, only because, yes, that information is forthcoming. It is going to be announced today, but the truth is, like, I have a phone call at 3:30 to understand all of the parameters about it, okay? Because I don't know the details, so I was not going to mention it to y'all until I had the deets. It is for sure happening. It is going to be an SU for whatever that's worth, so satisfactory, unsatisfactory. It is going to be an option. You know, one of my beefs when this was being discussed was if you're someone who's busted your hump and you've made an A or a B, you want that reflected on your transcript. Now, if you are someone who is in a class where you feel like you're underperforming and you're normally like an AB student, but maybe you're making a C in a class or something like that, then you would want the S on your transcript, right? That is coming. I have lots of questions and I'm happy to ask questions on your behalf. Some of the things that I'm about to ask this afternoon are things like, I believe these are not going to be factored into your GPA. So those are the kind of questions I have. If you end up with an unsatisfactory, at what point are we cutting off the U's? Because I think it's D's and F's and not just F's. Does that deny you credit for the course? You know, what are the pros and cons of making that decision? And so I feel like I will be talking to you guys either in a Zoom or I'll make a video with what I know about it. But I do know, based on what I'm hearing, that it's for sure your choice. You get to pick how you want to handle it. And the same with withdrawals. Anyone that wants to withdraw, you can withdraw up to the last day of class. You just have to let me know. So you're in charge of your fate for sure. I know that there's going to be a deadline for making a decision about your SU versus normal grading. But again, I don't have those details. So yes, it's coming. That's what I know. So
yeah, all the professor said was, like, later today you should receive notification about a pass-fail option for your current classes. I don't know the details,
okay.
but it appears that students will have the option. And, like,
yeah. Okay, so that's cool. So yeah, that's it. You guys probably have lots of questions. I do too. Now, if there are specific things that you already know off the top of your head you want me to ask on your behalf, I'm happy to do that. You know, I want to know about course credit. I want to know about, you know, many of you have PA or grad school aspirations. I want to know what people are perceiving as the pros and cons for you in that scenario. You know, my guess is that grad schools and PA schools, they're not ignorant of the fact that things are wild and wacky, but I have no sense of whether or not you would be more or less competitive if you had an S versus an A or a B. Personally, here's my 2 cents for purposes of our class. Now, we haven't had a lot of assessments, but I don't see any indication that y'all are performing less well than you were before we moved online. Most of y'all are excellent students by virtue of being upperclassmen, and also I happen to have a really packed and wonderful class. And so you guys are really good about communicating and getting things done. And so I don't see any reason that you guys need to fear that you're just going to fall off the end of the earth and tank and suddenly flunk the class or whatever. Because I'm going to work with you and you guys are communicating with me really well. So I don't see any reason you need to be fearful. But if you are someone who is teetering somewhere, then maybe you do want to consider it. I don't know. So those are, yeah. other university stuff that I know, I don't remember if this announcement was for sure certain the last time we spoke, but for sure, summer one is online. We are looking at possibly summer two, but then for the fall, who knows, question mark. So we know that. I got an email yesterday from my department asking me to approve, or there were several seniors that are slated to graduate in May, and several of those people are in this class. And I was asked, like, oh, do you think they're going to make it? And I was able to, like, resoundingly say, yeah, I reckon I'll let them pass and graduate. So at some point, I want us to maybe toward the end of class do something stupid, or I don't know, maybe I'll just wear a party hat or something that day, because it sucks for those of you that are graduating. Let's see, what else? That's, I guess, all the other university-wide announcements that I know of. All right, class stuff. Let's start out with your questions about Chapter 13 and or about the COVID lab. Carly, I know you had lab questions. Why don't we start with you, because I know you're locked and loaded.
Yeah, I'm pulling up the lab right now. Okay, they're basically just like, what specifically are you looking for kind of questions, because
Okay,
like
great.
I couldn't really find for the section 2, like, is there any list of symptoms that's unique to the coronavirus? Like, not really. Is that kind of what you were
Exactly.
looking for? Like, it's very okay.
Yeah, they're really nebulous, kind of not what we call non-specific symptoms. And so that really sort of, that question was intended. So I want you to like enumerate what those symptoms are. Tell me about the symptoms. What are they? If you've got the poops, does that mean you have the Rona? Spoiler, it does not, right? So what are the symptoms? And give me a little bit, not just like, oh yeah, the coughing and temperature, you know, da, 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 da. Give me a little bit like what happens in a more acute case. What is the symptom that seems to be causing problems for people where they end up in the ICU? Okay, so tell me those things. But that question is designed to lead you into this idea that part of the problem in this containment of this virus is that it's entirely reliant on our ability to test for it, because there's no one thing that we can put our finger on and say, aha, you got it. So we can't isolate it unless we know you have it. And we have done a miserable job of deploying those tests writ large. So that's kind of where that section is leading you, okay?
So then when the next question is like, which symptoms cause the most fatalities? I
Yeah,
know it's got a lot to do with the lungs, but I know the big thing I've been reading a lot is that it usually develops into pneumonia.
correct.
So is
So
that kind of like it's
that's
definitely like an attack on the respiratory system.
absolutely. So what happens is it seems to disproportionately affect your mucosa and your epithelium in your lungs. There seems to be a pretty disproportionate inflammatory response, which recruits things like neutrophils that go along, and killer T cells, and they come along and they eat up the lining of your lungs. And so what happens is you get this situation where your lung is broken, your lung lining begins to break down, and you begin to get fluid accrual in the lungs. And then that sets up a media for bacterial growth, right? So now bacteria can populate the lungs, but now you've got bacteria, you need to mount an immune response, and you're getting to a point of immune exhaustion where your body can't, it's sort of exhausted its immune reserves. And so you end up very quickly in a situation where there's a tipping point and you just kind of can't cope. So yeah, the other thing that happens is you end up with a fibrous or scarred lung as well. Like if you recover because of the amount of damage that's done to the lung lining, oftentimes you'll end up with scarring of the lung tissue. And the problem with that is, in order for the lung to function well, that tissue needs to be very thin and very moist, and that's how oxygen exchange occurs. If you have a buildup of fibrous scar tissue, then you have diminished lung capacity. You've lost the ability for oxygen to effectively diffuse across that membrane. So yeah, that's definitely the direction you should be going in with that question.
Okay. And then in the same section, I don't know if you're looking for like an exact, like, where it's like if Americans all stopped moving around and stayed in their houses today, how long before we would notice an effect? Like, you're looking for an exact, like, is there somewhere we can find that, or is it just
Okay, well, I'll give you a hint. That's going to be related to our thought on what the incubation period is for the virus. So if we know that it, typically symptoms manifest for X amount of time, and then you are sick for X amount of time, and then one of the things we don't know that you cannot factor into that answer is we're not exactly sure after you recover, are you still shedding virus or not? I'm hoping you've watched that MUSC Grand Rounds video, and they talk a little bit about the RNA coming off of people. There's a study looking at RNA coming off of people post-symptomatically, but it's hard to know if they're actually infectious or not, or if those are just random RNA particles, because that happens in a lot of other viral diseases as well, like the flu. So, but you're not contagious after the flu event is over. So we're still not exactly sure. Another thing so that's where that answer is going, is thinking about how long, incubation periods last. And there's no right answer. You could talk about incubation periods, but let's say that my family quarantines and we know let's say my husband goes to work today and brings home the virus today. And then he manifests symptoms in, a few days. And then he's got about a week of recovery, right? Well, depending on when or if any point during that time I catch it, then that's another lag time. And then if my daughter catches it, it's another lag time. So there's no right answer. I just kind of, again, this lab is designed for you to be thinking about these things. I want you to craft me a reasonable time frame and reason it out for me.
Okay. Okay, and then a question about, I know you went over it in the video, but when I go on this website, the DHEC website that has like the state by state and stuff information on it, that graph that you had, like the predicted cases for South Carolina, that's not popping up for me.
Ooh, have it taken off?
Yeah, it's gone. I rechecked because Carly texted me last night. I just rechecked it. It's gone. I have all the data if anyone wants it.
Okay, I can send out that data, but let me also see if I can find another more updated set of information for y'all, and I'll send that out because you have another week before it's due.
Yeah, because I was trying to do it last night, and I like went on the website and I found all the numbers for the first two parts. But for the second part, I was like, I was trying to read your numbers on the video, but they were kind of blurry. And
okay, well, let me do a little sleuthing. I'm gonna write down a note about that. Let me get you the SC numbers. I don't know if you guys saw in the state newspaper today, so the one from Columbia, right? This is an estimate because we don't have good testing data, but they estimate that we're probably at somewhere around 16,000 cases in South Carolina, which is not going to be reflected in your data that I get for you. I may be able to get it like through the hubster. He might have access to that data that I don't have access to. That's not going to be reflected in what I send you. So these are estimates based off of epidemiology and things like that. So what you got, Cheyenne? Were you going to say something?
Yeah, so I submitted my lab report to you the day after you posted the video, but because we talked online, I didn't realize that there were other things I had to change, so I went back and changed the thing. So should I just wait to resubmit it again? Because I changed
Yes, because I actually I'm working on going through yours. There's a couple places I want you to go back and recheck. I think there's a typo. At one point you told me the flu killed 15% of people. I think you meant 1.5%. Because 15% would sure enough be catastrophic.
Yeah.
So there were a couple places, and there was another thing, and I'm glad you brought that up because I wanted to mention this. I haven't gone through every single resource that Professor Bauer linked for you guys. I've been doing my own research, and so I kind of knew the answers to the questions. I don't know if there is some confusion over ventilator masks versus ventilators. Okay, so like Cheyenne had some very big numbers. She was like, oh, there's this many ventilator masks. That's just the piece that goes over your mouth. The number of ventilator units is far, 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 far lower than that. That question was intended for you guys to understand that very quickly we can exceed our healthcare capacity. So I don't know if that was listed somewhere and confusing in the resources. Cheyenne, what?
That is now on the SC DHEC website. It has like beds and masks and stuff.
Beautiful. Okay.
So
So,
I can fix that. No
yeah,
problem.
do you see the difference? Like
Yeah.
the number of actual ventilator machines is like muy poquito. I mean, which makes sense because they're really, really expensive. And actually NPR did a survey looking at where regions that have the highest number of ventilators per capita. And Florence actually made the list of having a lot of ventilators per capita, one of the best numbers in the nation. But here's the caveat that NPR doesn't realize about Florence, South Carolina. I want to say it was somewhere in the ballpark of 60, but I could definitely be wrong because I have a lot of numbers in my brain right now. What NPR doesn't realize is that Florence doesn't just service Florence, right? If you look at the population of Florence as per like the census data within the city limits, we're kind of a tiny town, right? But if you look at the region, like essentially Florence is healthcare for the entire PD region and into the Piedmont of North Carolina. Like my husband sees North Carolina patients all the time. So it's kind of misrepresentative. If there's like X number of ventilators in Florence, that's because we're a regional referral center. Yeah.
Okay, so I'm looking at the DHEC website for the ventilators, but this graph is for invasive ventilators needed. Is that what you're looking at, Cheyenne?
Needed, the ones we need.
Yeah, this is like a projected, the numbers that they think we'll need per day.
Okay, let me.
The website that I found said that we had the South Carolina at the date that it was written had about 1,200.
Okay.
Or something like that. So
okay.
I don't know.
Okay, I'll double check that number for you too. Okay, I'll see if I can't find a good resource for that because I wasn't sure based off of Cheyenne's response. I was like, I need to go through all of Dr. Bauer's links. Now you're not limited to, don't just stop with the links that are on there. But I wondered if there was something that was misleading in there because that didn't seem right to me. So
no, I started going crazy because I was like, I don't know where to find any of this information. So I started doing like weird math that I pulled from websites and then I just made things happen. Same for like one of the how many beds. I was like, let's just estimate there's 50 beds per hospital.
No.
Now they have that on the website too, where you can follow that.
And I have a link to how many beds there are in South Carolina. So
yeah, because whatever article I found said that there was 1,260 as of March 25th, 2020 in the state. I don't know. Yeah.
It seems high to me. I'll double check that.
So I was like, I don't know. I don't remember where I found it.
Maybe, but that does seem kind of high to me. So I'll double check that.
Okay.
And one thing that you guys may want to do, I will not ding you if you accidentally stumbled upon an erroneous number, but what you might want to do to help me go back and go, where did this come from? If there's something like that, just include the link. And then I can go back and be like, I wonder where that came from. Because it's not your fault if somebody puts that information out there, right? Unless you're going to something like, theinquirer.com or, somebody's TikTok page, then that's on you. But so yeah, you may want to link where you're getting things from. One of the things that I told Cheyenne, and I didn't mention in that video, one way to get at the rate of when you're doing the increases is you can actually graph the data and get do a linear regression and get a slope of a line, and that's the rate. The reason I didn't bring it up is it depends on which data you grab. You want to grab data that appears to be linear because remember, this stuff is increasing exponentially. And if you do a line graph, you're getting a line and that's going to give you a little bit of an erroneous number and actually a little bit of an underestimation. But if you get the rate that way, that's fine with me too. I'm looking for ballpark ideas of how quickly it's, growing.
Yeah, because I had a question about that too. So are we supposed to do that for the day by day that we got as well?
What do you mean?
Because like in the video, you told us to do like week 2 divided by week 1, but that was for the South Carolina cases.
Yes.
We need to do that for the 14-day data as well.
Yes.
I did that and all of mine are like 1.2, 1.1. So then what do I do with those numbers from then?
So for each day, but what you want to do is figure out how many days it takes to double. So okay. You're looking at the doubling rate. So don't necessarily look day by day. Look at, okay, so
first day.
Yeah.
And then find
the doubling point. So that was however many. And then what's the next doubling point? And what's the next doubling point?
Okay. So I didn't have to do the whole one, like divide the second day by the first day thing for
No,
this?
the only reason I did it that way for the week data was because you got 7 days in between and you were doubling within that time. So each data point for South Carolina doubled, more than doubled, because we were looking at it a week later.
Okay.
So
Gotcha.
I was looking for when I could find that doubling. So you can actually go through and manually look for, so it's probably like every 3 days or something like that, based on what you're telling me, but yeah. Related to lab, not this one. So, you know, Thursday would be our normal lab day. I'm going to go ahead and post your next lab. But again, what I've decided to do is this lab, this COVID lab, is going to count twice. So it's going to be 40 points. All right. The CRISPR lab that I'm going to post for you guys is also going to be 40 points, and it'll be due at the very end of the term. Okay. So you've got 2 weeks for each of them. And I think that's going to help everybody. That way we're not posting a million things and you guys can just work through these, you know, as you need to. But I'm going to go ahead and post it tomorrow so that you have it and you can start working on it at your own rate. Okay. And while I'm on things that I owe you, I have recorded my chapter 14 lecture 4 different times now. I managed to either say the F-bomb, have a catwalk across the screen, drop something, or completely mess up in some way. So I should have that posted for you right after class. Sans F words, sans kitty cats, okay? Doing the best I can. I don't know about y'all, but the fatigue of this setup is starting to set in and I find myself making a lot of mistakes. So I'm catching up. And I know I owe you a chapter 14. We're going to do chapter 14 in one lecture. I'm actually really reducing what I'm asking from chapter 14 because I'm starting to triage what we're going to talk about so we can get through the high points that I want you to have before we leave class. Okay. What else? More questions about the lab or chapter 13?
What is a live virus scan?
Okay. So that you kind of had to interpret from the MUSC video. There's a couple different ways you can estimate someone's infection rate. The way that they're doing it and the way, like if you went in today to get tested, would be a PCR test, right? And you would run PCR and you would just use that PCR data to measure how much of the viral RNA you have. And you would use that to base an estimate of what your viral load is. So you look for presence or absence of the viral RNA to tell if you have it. And then you would look at how much of the RNA is to know what your viral load is. The problem with that is sometimes that RNA can be present, but it's not actually formed into an active virus with a lipid membrane and the glycoprotein spikes and the whole bit. So if it's not formed into the active virus, then you don't have an infectious unit. It can't get into cells, right? The live virus scan, what they do with that is they basically swab you and then they try to provide it with a cell-like food, cellular food that it can infect. And then how much it infects that cell culture gives the practitioners some idea of how infectious you are. Does that make sense? So while the PCR test can tell us if you have it or not, it can't tell us what your viral load is, and it can't tell us whether or not you are actively infectious. That's the problem. So live virus scan is, I don't really like that term because viruses are not alive, but it's sort of like their ability live in that they're kind of hot. That's their ability to infect. Okay. Does that make sense?
Yeah.
Okay. What else?
Okay, I had a question about the paper.
Yeah. What you got?
I'm stressed about that.
Okay. Let's talk about it.
When is it due? What's the exact due date?
Okay. I had the due date as, let me pull it up. I think I moved it to like April, the very last day of classes, which I believe is April the 27th. Does that sound right to you guys? I'm pulling up my, does that sound right to everybody else? Let's see here. Course plan post-COVID. Okay, no, actually, so what I have, what I have right now, and I can be flexible about, and let's just talk about it. I've got like 47 windows that just popped up. Okay, so what I have on my screen is that the paper was due on Friday, the 24th of April. That was the last full week of class. And then that following week, Monday, the 27th would be our last day of class. And I have a homework that's due that day. That's going to kind of be your like last little homework for me. And then the 28th is reading day. And then the next time you guys would hear from me would be at the final. So tell me what's stressing you. Like, we can move the due date. I don't want to move it into exam week because I feel like that would really kick your behind in a big fat hairy way.
Just like, I don't know. I'm just looking at my piles of work. Oh God.
Okay. So when I initially assigned the paper, I think I told you guys it needed to be 5 pages. Is that what I told you? I've got it right here too.
That's 1,250 words on the paper.
Yeah.
I think that's roughly 5 pages.
Yeah. So, you know, at this point, you know, in the past I would have said, that's 5 pages is your minimum, blah, 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 blah. Please exceed that. I would say don't exceed that. Make, you know, you've already probably got plenty of information for 5 pages. Carly, do you have the rubric in front of you, or my instruction sheet in front of you? Excellent. How many references did I say I wanted?
8.
8. Okay, if that becomes an impediment for you, then that for sure is something I can relax, because I do understand that doing research is a little more difficult now, okay? So if you're like, I've got these 4 or 5 papers and I feel like they really cover the material and I feel good about it, I'm okay with that. All right, so I'm fine with relaxing that component. What else would help?
Oh, so when is our final exam? I don't have it written in my planner for some reason. Does anybody know off the top of their head?
Hold on. I had it, I think, I want to say, I'm going to the registrar page because I had it written down somewhere, but I don't remember where. Oops. So while I'm typing and while we're on this topic of people feeling overwhelmed about work, I've been thinking about that. Everything is fluid because I've never done this before where I had to change classes. I've never taught an online class. I've always taught in-person classes, and I've never had to on the fly change everything. And I feel like if you were an online student, your course load would be different than you designed it to be as a face-to-face student, right? So I'm trying to like be very flexible about that. And so along those lines, while I'm looking this up, you know, initially I was like, well, I'm just going to give them a take-home exam and it's going to be these essay questions and they're going to respond and it's going to be open note and open resource. And we can still do that. That would be another assignment to add to your ever-growing pile of assignments. And so my question for you is and don't answer it right now because I think I'm going to send out a poll. I'm going to send one of those doodle poll links out. But here is what I'm also thinking could be an alternative. I think that another way we can go is I can post an exam in Blackboard that's structured like your previous 2 have been, where there would be a series of multiple choice and short answer questions. And then I would time it and I would just give it a little longer than we would in class, like 90 minutes, so that you study up ahead of time. Now, you know, you're at home, you'll have the ability to look some stuff up, but you would also have to do some studying ahead of time or you would run out of time, right? So I'd give you like 90 minutes to complete, and I would make it exactly the same way that I have been making the exams. And my thought process there, and y'all think about this, I'll send out a doodle poll. We can do it however you want. But my thought process is in that scenario, you're constrained. It's not this lingering long ass assignment that you guys add to a series of lingering long assignments that have been given to you. So think about that. Think about that exam structure. Would you rather have this open resource, sort of longer written out exam that's going to be a series where I'm going to kind of expect you to have these, you know, longer, more detailed responses? Or would you rather have something similar to what we've been having, given the fact that you still have the 2 labs from me and you've got your paper? Okay, so think about that exam structure. All right, and then final exam is boop, boop, boop. I'm pulling it up. When do we have class? All right, we have class on, let's see here. Wednesday the 29th is the first day. Oof, are we May 5th? No, that can't be right. Hold on. Monday, Wednesday, Friday.
I have the first written down.
Yep, that's it. I just found it. So okay, yeah, our final exam is scheduled on Friday, May 1st. And here's what I will do with that for sure. For sure, that's just gonna be multiple choice. For sure, that'll be online. For sure, it'll be, I usually double the number of questions. So if I give you 50 points, then it'll be 100 points, but I'll give you like the full amount of time, which is like 11:45 to 1:45, so that's 2 hours. I'll give you 2 hours to take it. And here's what I'll do. I'll just set it for 2 hours. I don't care what time you hop on to take it. It's just at the end of 2 hours, it's done. So if you want to hop on at 5 p.m., if you want to hop on at 1, you know, because I know you guys probably got brothers and sisters and cats and dogs, and if your sleep schedule's probably whack-a-doodle-doo, mine is too. So I'll just turn that bad cat on. And these are going to be questions that either you've seen before or they're simpler than that. Some of my multiple choice questions can be hard, right? So I will be generating a study guide for you. The study guide will be like, these are the topics I'm going to cover on the exam. Okay, because we can't possibly cover everything on the exam. So you will get that from me, and you'll get that from me probably like that second to like in 2 weeks. So yeah. What else?
I also wanted to ask what we're going to do about our lab notebook grade.
So lab notebook grade is, that's the whole reason you're doing these labs. So the lab notebook grade is what I've been taking up all along, right? The 20, 20, 20, 20 points. So you guys already had that hay in the barn. And then by virtue of turning in the assignments that you're turning in for the online stuff, that should equate to the whole lab notebook grade. Let me double check that I'm not lying to you. So that should take care of itself, hopefully. Let me just double check again that I'm not fibbing. Did I have a separate? There we go. Syllabi. Oops, that's the wrong one. So stop it. Oh, I see. I had like a separate lab notebook for 20 points, just having you guys turn in everything collated all together. I'll probably just dump that grade, y'all. I mean, I don't see how you could possibly get that back to me. And you know, don't worry about it. Yeah. What else? I did have the exam pulled up. It's been a hot minute since we did that. I can talk about some of the things that were on the exam that folks missed if you'd like me to go over that. Devin's doing a nod. Okay. So I'm going to share my screen really quickly. I've got 47 windows pulled up. There we go. Can y'all see the exam? Okay, so most of y'all did pretty well with the first page. You guys were just really on top of it. Hold on, I'm grabbing my notes. The questions that sort of outlined the various mutations and translocations and all that good stuff, you were on top of that. Where you guys started to get a little bit off was on the second page. Hold on, let me pull up my notes. There we go. Okay, so first page, I think everybody mostly got this right. So like, which type of chromosome mutation increases all of the chromosomes? You guys knew that was polyploidy. Which type of chromosome mutation decreases the amount of genetic material? You guys knew that was a deletion. Chromosome duplications resulting in abnormal phenotypes because Developmental processes depend on relative amount of proteins. Does all this sound familiar to you guys so far? Yeah? So you guys did pretty well. Let's see here. On all of these, you got, like, when I showed you the rearrangements, you were good. Kind of don't want to go through all these because you mostly had them. Now let's see this one. Okay. Number 6 kind of tripped a couple people up, and I think it was just, okay, let me just read it. And it's a vocabulary issue. So the unique and elusive organism, Muhammad Dawadii, has 2n equals 38 chromosomes. How many chromosomes will be found in a trisomic member of this species? So a trisomy is when you have 3 copies of one of them. So the answer is going to be 39, because you're going to end up with 1 extra because you got 3 copies of one of them. And so some of y'all fell for my, I think I doubled it. Yeah. And so, or something like that. You guys fell for a couple of these. So, but the answer was 39 because a trisomy produces 1 extra, because you'll end up with 3 copies. Like if you had trisomy 21, you'd have 3 copies of 21 when you only should have had 2. So you've only got 1 extra chromosome. Does that make sense? Let's see, that was one that a couple people had trouble with. But everything else was good. Let's see here. I'm trying to think of ones that you guys missed often. Okay, let me go to the second page here and tell you about a couple that you guys missed often. So I'm going to show you, start here. The fundamental repeating unit of chromatin is nucleosomes, guys, because we're talking about chromatin, not just DNA. So some folks fell for the nucleotides answer because they're like, oh yeah, it's fundamental repeating unit of DNA. But chromatin is when you have DNA plus proteins. And so the fundamental repeating unit of that is nucleosomes, okay? A lot of you guys knew that if you acetylate histones, then what you're doing is you are neutralizing the charge, which means that the histones will not bind to the DNA as well, and that would increase DNA transcription, okay? Let's see here. Most of you guys got this one right about which was not true of heterochromatin. Remember that heterochromatin is the chromatin that's super duper condensed. And so the one that was not true is this one here. It makes up most chromosomal material and is where most transcription occurs. No, that's the opposite is true of that, okay? Let's see here. This one was one that y'all missed a lot. Let me go through and read it. Okay, so which of the following statements is not true? Both mitochondria and the chloroplast generate ATP. That's true. A single eukaryotic cell may contain thousands of copies of the mitochondrial genome. That is true. According to the endosymbiotic theory, chloroplasts are thought to have evolved from cyanobacteria. That's also true. The mutation rate of mitochondrial DNA is higher than the mutation rate of nuclear DNA. Also true. The one that was not true is this one. Oxidative phosphorylation capacity is constant throughout a person's lifetime. It diminishes over time. And I believe we talked about that. Let's see here. Oh, this one was missed a lot, and I think you'll see why the minute I point it out. How many different types of histones are found in the nucleosome that packages mitochondrial DNA? It's kind of a trick question. There's no nucleosomes in mitochondrial DNA, so it should be 0. So yeah, it was a little bit of a trick question. After I wrote that and then did it, I was like, you know, that was kind of a jerk move. So, you know, it's one of those things where I know that that's built into that. So when I look at your grades and you're like a few points away from something, I'll be generous because that wasn't a great question. Let's see. I think most of the rest of this stuff you guys did really well. Where people started having trouble was with some of the short answer stuff, okay? So in describing PCR for me, I did correct that. Remember in class, I told you guys it was 3 steps. So you needed to tell me something about the denaturing step, the annealing step, and then the elongation step. Those are the 3 things that make up PCR. You needed to tell me about the ingredients, which included things like DNTPs, right? You needed to tell me about the fact you needed a template of DNA. You needed to tell me that you needed a DNA polymerase enzyme. So those were the things that I was looking for to make that a complete answer. It wasn't important to me that you knew the exact temperatures that things happen, just sort of that you understood you heat it up and it comes apart. And oh, primer, I forgot to include that. You cool it down and the primer anneals, and then the polymerase attaches and it elongates when you make it kind of an intermediate temperature. That's kind of what I was looking for, okay? You guys did really well on number 2. You knew that Sanger DNA sequencing differed from traditional PCR and that it had those dideoxynucleotides that got put in that terminated the reaction. And so you could use that to like have all these little fragments of DNA that just kind of got longer and longer and longer. And then you can read those through electrophoresis. Number 3 had to do with the fact there were 2 things that you could say on number 3. So number 3 reads, briefly explain why in humans and other mammals, sex-linked aneuploidies are more common than autosomal aneuploidies. And so we talked about that. That had to do with the fact that in mammals, 1, you have a dosage compensation mechanism in females so that only 1 X chromosome is active and other extra material gets shut down. So that helps to mute those effects in females. In males, especially in human males, the Y chromosome doesn't have many genes on it. So if you ended up with duplication of that, you wouldn't necessarily be in big, big trouble. So those are the reasons for that. On this one, we discussed 2 ways that Down syndrome could arise, describe these 2 mechanisms. That was the trisomy 21 was 1. And then you guys remember talking about the translocation where you can have a wee little piece of chromosome 21 that ends up on chromosome 14. And I think like a lot of folks got that. Let's see, I had the other stuff in a separate document, so I'm just gonna read those to you guys. I asked about the Hershey-Chase experiments, and pretty much if you guys told me about it, that was great. A lot of you guys kind of got confused among the different experiments, so I tried to give partial credit where I could. You guys did great. The next one is about nucleosomes, and I asked you to diagram it for me and tell me the different histones. You guys did fantastic on that. One of the things that you guys struggled with was that there was a diagram where you had to label stuff. Let me go ahead and find that and pull it up. Because that one messed several people up. And I'm not exactly sure why. Maybe you guys can help me so that I can fix that question in the future. Let's see here. It was under exam 2, part 2. There we go. Okay, so let me share that document with you guys. Hold on. All right, can y'all see that now? Okay. This diagram right here is the one that tripped a lot of people up. So I asked you guys to kind of label it with the appropriate enzyme or structure, okay? So this right here is referring to this little piece that's at the beginning, and then you have another little bit that's being synthesized. And so what I was hoping you would deduce is that this must be the RNA primer that's needed to get that started, okay? Then you can see that this right here seems to be making a new DNA molecule. So this would be DNA polymerase, okay? And if you told me which one, that's great. But if you just put DNA polymerase, I accepted that. All right. Then let's see here. These were the single-stranded binding proteins. A lot of you guys got that one, okay? This one is helicase and this one is topoisomerase, and those were often confused, and I tried to be generous on that because I can kind of see how you got there. But if you look really closely, you can tell that this is relieving the twisting forces while this is kind of pushing the double helix apart, okay? And then is this the leading or the lagging strand, and how do you know? I think almost everybody got that it was the leading strand because you're making 5 to 3' and it's going continuously. So I think everybody got that. And then the last 2 had to do with telomerase. So I asked you to describe for me the end replication problem and what effect it has. So you guys did really good job telling me that basically because you need this primer, you can't get to the very end of the molecule. And so it leaves a little bit that over time degrades. So basically your telomeres degrade and gets shorter and shorter as you live. But there is an enzyme that takes care of that, which was number 11, and that's telomerase. And you guys told me a little bit about how it worked and where you find it. So if you remember, it's sort of BYOP. You guys remembered that phrase. It brought its own primer, and it used that to sort of synthesize a little bit of extra at the end that could then fill in that gap. And you found it in stem cells and cancer cells, weirdly, okay? Seems to be related to cell proliferation. So, let's stop that share. So that's that. Any questions on the exam? If you have a question about your personal grade, then let's just do like a private Zoom or something like that, if you need me to go over that with you. And like I said, that there was one question and like maybe two where I was like, I didn't love it. I didn't curve this exam, but I kind of know in my head, like there's a couple of points out there that I'm feeling generous about. So like when you're on the precipice of needing me to do you a solid because you have a 78 at the end of the semester or something like that, I'm gonna bear in mind that there were some crunchier exam questions, okay? So just trust me to do right by you. What else? So my agenda for the day was to mention the lab, to talk, ask if there was any questions about chapter 3, go over the exam with you, mention our last exam and how you guys may want that formatted. And so those were the things I wanted to talk about. What I have on my list of things that I need to do for you, this is homework for me. So my homework for you is, number 1, I'm gonna get you guys a couple of links in addition to the ones that you have to help you complete your COVID lab, including some new and better South Carolina numbers. And I'm gonna look up and double check the ventilator numbers for you guys, okay? I'm also gonna be posting my 5th iteration of the chapter 14 lecture, and then I'll have the chapter 15 lecture up for you guys. I think that one will probably be a couple of lectures long. So I'll have for sure one of them posted by Friday, so we can get back on track with our lecture schedule. I'll also have some more homework questions posted by Friday, maybe by tomorrow, so you guys can have the weekend to work on that. And I'm going to send you a doodle poll. That's my last point here, to ask you what your preference is for the final exam format. Is there anything else I should be doing that I'm forgetting that I need to do for you guys? All right. And look, I'm glad that Hannah brought up that there's some stress over the paper. So, you know, let's just continue to talk about that. If you're getting, like I said, a few resources and you're getting 4-ish pages in, not quite 5, and you want to shoot it to me and be like, hey, I think I got this. I don't want you turning it into a 2- or 3-page paper. But if you want to, we will adjust as needed. And if you need an extra day or 2 to get me that paper, we can adjust as needed, okay? So I don't want you guys, I mean, we're all stressed out. I don't know about you guys, but it's not fun anymore. Not that it was ever fun, but we're starting to kind of see we're in for the marathon and not the sprint. And I'm starting to question what's going to happen when I run out of toilet paper. So I get it. I do get it. Nothing else from you guys? Okay, well then I'm gonna go ahead and end this and post this for folks that weren't able to make it. You guys know, if you need anything, we can definitely, like Cheyenne and I Zoomed the other day, she had a couple questions. It was really effective way for me to answer questions. So the best way for us to arrange that, if you need it, is just shoot me an email, like, hey, are you available on Tuesday afternoon or something like that? And then we can arrange a time for us to virtually get together individually, okay? All right, guys, be well, and just let me know how you're doing. All right, have a good week or rest of your week.